Hi everyone, I am ZHANG Yue from Qingdao, #2 Middle School of Shandong Province, China. I'm very glad to have this chance to share my works with you. The title of my project is Screening of Tumor Drug Resistance Genes by Bioinformatics, EGFR targeting drugs as an example. This is my outline. First, why I do this work? When I was a little boy, I loved animals. I loved them because they are beautiful, amazing, and full of changes. I appreciate the beauty of nature and the variety of species. When I entered the lab for the first time and watched under the microscope, I saw this, just the cells of lung cancer. And as you know, cancer has become a major threat to human health, killing millions of people per year. At that time, I began to realize that life is not just about its beauty. Life scientists must take responsibility to protect the beauty of lives. This figure is from Cancer Statistics 2020. It can be found that the death rates for lung, colorectal, breast, prostate cancer declines, and the declines for lung cancer accelerated. However, lung cancer still caused more deaths in 2017 than breast, prostate, colorectal, and brain cancers combined. EGFR, epidermal growth factor receptor, is a transmembrane glycoprotein. It belongs to a family of 4 related tyrosine kinase receptors and acts as a key mediator in cells signaling pathways involving cell proliferation, apoptosis, angiogenesis, and metastatic spread. The overexpression of EGFR in a variety of human tumors and the critical role of the EGFR in the cell signaling pathways makes it a primary target in cancer therapy. EGFR TKIs such as gefitinib showed high significant sensitivity for the treatment of patients with EGFR mutations such as L858R. And they are recommended by international guidelines as a first-line treatment regarding the higher efficacy and safety. Now, there are 4 generations of EGFR TKIs that are used in first-line treatment. Despite the excellent disease control with initial EGFR TKI therapy, acquired resistance is ubiquitous and remains a key challenge. The first generation of EGFR TKIs, such as gefitinib and alertinib, exhibit positive clinical responses between 50 and 80% of the patients who are responding. These patients with EGFR mutations. However, these patients who are responding to these drugs have faced secondary drug resistance after only 1 year of treatment, dramatically limiting the efficacy. The EGFR gatekeeper T790M mutation, also known as the primary mechanism of the drug resistance against first and second generation of EGFR TKIs, led to the discovery and the development of third generation of TKIs. Soon, new mutations such as C797S happened, and new TKIs have been introduced, and the 4th generations of EGFR TKIs have been introduced. Actually, multiple pathways have the potentials to blunt efficacy and cause drug resistance. The multi-target agents and combination therapies are 2 appealing mechanisms to both overcome diverse resistance and enhance the efficacy of EGFR TKIs monotherapy. Molecular response profiling and drug screening should be used to investigate early adaptive responses after EGFR inhibition and identify candidate EGFR TKI escape mechanisms and combination therapy targets. In this paper, Differential genes associated with drug resistance to gevtotinib were screened by bioinformatics methods, and we hope to provide some help for exploring the candidate EGFR TKI escape mechanisms and finding the new combination therapy targets. Second, methods and results. 36 samples of non-small cell lung cancer obtained from GEO database were divided into treatment and non-treatment, drug resistance and non-resistant group. And the GEO2R was used to extract the differential expression genes. For the data from the accession GSE 75309, when the logarithm of fold change on the base 2 is larger than 1, it is chosen as a threshold criteria. 33 genes such as SIDE2, TAGLN have been found. For the data from the accession GSE 83666 with the same criteria, 86 genes, including SIDE2, LOX, GOP, KRT17, and so on, have been found. Notably, the gene SIDE2 show high difference expression between both treatment and non-treatment, drug resistance and non-resistant group, which may indicate its special significance in the study of gevtotinib resistance. SIDE2 is ubiquitously expressed protein exhibiting a high affinity for the ZH1 domain of the transcriptional co-activator CBP-P300, also known as TAZ1. The ZH1 domain of P300 also binds hypoxia-inducible factors 1 alpha, also known as HIF1 alpha, and some other transcription factors. Due to the interactions with CBP-P300, SIDE2 was reported to act both as a positive and a negative regulator of transcription. It appears to depend on whether SIDE2 enables the interactions between CBP-P300 and other transcription factors, or it prevents such interactions. The interactions between HIF1 alpha and TAZ1 under hypoxia conditions are related to transcriptional regulation of genes that are crucial for cancer cell survival. The role of SIDE2 in the inhibition of HIF1 alpha dependent transcription and the maintenance of cancer stem cells reveals its potential as a target in NSCLC. Past drug candidates have tried to interpret the cancer cell survival mode by stopping HIF1 alpha from ever reaching TAZ1, but they haven't been very effective. In recent work, scientists found that it may be more efficient to mimic nature's own response and use a SIDE2-like drug to knock HIF1 alpha away. Despite the evidence linking both SIDE2 and HIF function independently to several aspects of cancer, little data linking this interplay between these two factors is available. Therefore, comprehensive studies are needed to develop innovative targeted therapies. 3. Conclusions and future. Based on the screening of differential expression genes, some possible therapeutic targets such as SIDE2 for the treatment of cancer were proposed. And as cancer cells continue to mutate, we have to continue to develop new drugs. It is like a circle without an ending. So I'm thinking that should we find some new ways to get out of this circle? A SIDE2-like drug or new combination therapy targets should be considered to develop for catching up with the speed of cancer cell mutation, in my opinion. Of course, further studies such as screening on more examples or new methods and the experiments should be done in the future. Thanks for listening. Thank you.
Hello.
Hello.
It's a very interesting theme. Thank you for this project. Say me, please, what software did you use for screening?
The software. The first, I just used some database. The first database I used was the GDSC database. On that database, I found that EGFR is very frequent in cancer mutations. So I think I chose EGFR as my target. The second database I used was the GO database. On that database, I got two accessions. GSE 75309 and GSE 8366. I divided genes into different groups and used GO2R to extract the differential expression genes and got my results.
Okay, thank you. But maybe you use programs for modeling biological activity and toxicity compounds. It may be important for especially for synthetic chemists. No, don't use.
Yes. Excuse me, would you please ask the question again?
What program do you know for modeling biological activity and toxicity for compounds?
Problems.
Maybe PASS or QSAR. Don't use.
I think some problems. I think some problems is that the EGFR TKIs is very effective, but the drug resistance will eventually developed after nearly 1 year of treatment. That not only did it dramatically limiting its efficiency, and we have to continue to develop new drugs. So that this will make the drugs of these TKIs very high. So and makes many patients cannot afford this. So I think we should find some new ways or use some combination therapy targets to treat this cancer. Maybe we can get better results.
Okay, thank you. It is very big research. Thank you.
Thank you.
I agree with David. It's quite a large research, and I have one question. Can you please tell us just in 2 words what is your own contribution to this work? Do you understand my question?
Yes. I think that I think I used the bioinformatics methods. Because of the COVID-19 epidemic, I have no chance to go to the laboratory to do any research. So I just used bioinformatics to use bioinformatics methods to to find some data find some data and divided them into groups and extract the differential expression genes and got my results.